Welcome to the Dana Parish Podcast, Third Opinion. Today, I welcome molecular biologist Professor Richard Ebright. He is the leading authority on gain-of-function research and has been warning about its dangers for decades. We discuss the shocking historical timeline, the players, the press, and the cover-up that gives context to how reckless science likely led to the COVID-19 pandemic. Drop me a comment, and please don't forget to subscribe.
Great to see you, Richard. I really appreciate you doing this with me today.
Thank you, Dana.
You are a highly distinguished scientist. You are a professor of chemistry at Rutgers. You have so many accolades that we could spend the next half hour or an hour going through them. But one of the things that has struck me about you that is so remarkable is your decades-long advocacy to put parameters around dangerous bioweapons, gain-of-function research. In 2004, I found this great article about you in the New York Times titled, "I Beg to Differ, in a Lonely Stand, a Scientist Takes on National Security Dogma." And in this article, you were coming out against the proliferation of these very high-level biosafety labs, biosafety 3 and 4, and very dangerous experiments can be conducted at these labs. And you said, "I'm concerned about these labs from the standpoint of science, safety, security, public health, and economics. They lose on all counts. I think there's a very real threat of bioweapons use." Is this pandemic, the COVID pandemic, the kind of thing that you were warning about then?
Yes, this is exactly what we were discussing at that time.
I reached out to a bunch of scientists before we did this interview to say, if you can ask Professor Ebright, one question, what would it be? 3 people said to me, how angry is he after warning about this for so many decades and in such detail and so relentlessly?
I think the relevant emotion would be disappointment and dismay rather than anger. The COVID-19 pandemic appears to have emerged from a research incident from a laboratory. If that can be verified, it will have been the most significant industrial or research accident that has occurred in human history. It has so far, and just so far, cost more than 20 million lives, and it would have been entirely preventable. It was foreseen in detail, down to the identification of the specific research project and the specific location that was most likely to result in a laboratory-generated outbreak, in a laboratory-generated pandemic. Had warnings been heeded, warnings that were repeated again and again over the past 2 decades, this would not have occurred.
Who didn't heed these warnings?
The person who played the single most important role in the United States and outside the United States in enabling, encouraging, and excusing incidents of the kind of reckless research that likely gave rise to the pandemic was the former National Institute of Allergy and Infectious Diseases Director, Anthony Fauci.
What was his role specifically?
Early, and by early, I mean as early as 2001 and 2002, in the reaction to the 9/11 terror attacks and the anthrax mailings, which occurred in the United States at the same approximate time as the 9/11 terror attacks, a decision was made to appropriate funds. Large appropriations were made for expanded biodefense. And early in the process, Anthony Fauci was assigned key responsibility for leading U.S. biodefense efforts, not just within his organization, not just within the National Institutes of Health, the NIH more broadly, but across the entire U.S. government. He was assigned as, in essence, the biodefense research czar. He saw the best response to biodefense concerns as an expansion of research on biological weapons agents. That is to say, on fully infectious, fully virulent pathogens and biological toxins that could be used as potential weapons of war or instruments of terror or agents in a biocrime. And he argued that the appropriate response was to expand U.S. research, dramatically expand U.S. research involving fully infectious bioweapons agents. In his view, the appropriate reaction was a large-scale expansion of U.S. research with biological weapons agents per se, research handling the agents. This was accompanied by a large expansion of infrastructure for the research, large numbers of high containment laboratories, a very large expansion in biocontainment laboratory capacity, a very large expansion in the number of researchers and the number of research. Some scientists and many science policy specialists at the time were concerned about this, seeing this as precisely the wrong response to the anthrax mailings. Very early on, it became clear that the anthrax mailings were not an external attack by an external adversary, but were perpetrated by a person from within the U.S. biodefense research sector, a person who had authorized access to anthrax spores and then released the anthrax spores. And many were concerned that expanding the number of institutions and individuals who would have access to biological weapons agents, such as anthrax spores, and doing so without a concomitant increase in the level of security and oversight of that research, inevitably would result in accidental and deliberate releases. In other words, that the expansion of biodefense research with actual contact, possession and handling and work with these agents would increase rather than decrease the risk to the American public.
So did the U.S. use that in the sense of, never waste a crisis? Did they use anthrax to expand all of these?
Oh, absolutely. So there was a direct reaction to the 9/11 and the anthrax mailings that resulted in a very large increase in appropriations for biodefense research. This occurred across multiple agencies. It occurred across the Department of Defense, the Department of Homeland Security, the United States Department of Agriculture for plant and livestock threats, the National Institutes of Health, the CDC, etc. All of those agencies received large appropriations. The initial quantities of the appropriations were so large that they could be absorbed and used only by infrastructure projects, which is to say construction, large-scale construction and expansion of high-level biocontainment laboratories. The sustained funding after the initial burst of funding in 2001 and 2002 continued to operate those newly opened laboratories and then to fill those laboratories with newly hired researchers who would carry out research on biological weapons agents. And the person who was placed in overall authority over this entire program, which, as I just told you, spanned multiple agencies, was Anthony Fauci. He was selected by then Vice President Dick Cheney. Dick Cheney believed that the United States should adopt a more assertive and aggressive biodefense posture and should carry out research, even if that research approached red lines set by the Biological Weapons Convention, the international treaty banning production of bioweapons and use of bioweapons. He felt that the United States should perform research that approached those red lines and even in some cases reach those red lines and even in some cases cross those red lines. He was very concerned that the Department of Defense was not carrying out an aggressive program of research that would approach, reach, and cross red lines, and it was not doing so because the Department of Defense, while carrying out research on biological weapons agents, maintained a BWC, Biological Weapons Convention Compliance Office, that would review each research proposal. And he was concerned that that BWC Compliance Office was too restrictive and would stop proposals that approached red lines. And so he sought a new agency and a new authority that could carry out the research with no BWC compliance.
That's huge. That's really important to know.
And he found that the Department of Health and Human Services did not have a BWC compliance office. The National Institutes of Health did not have a BWC compliance office. And the institute that Fauci ran within the National Institutes of Health, the National Institute for Allergy and Infectious Diseases, did not have a BWC compliance office. Therefore, Cheney transferred all overall oversight authority across agencies in the United States government, and most of the funding out of the organizations like the Department of Defense that had been engaged in the research previously and into the National Institute for Allergy and Infectious Disease with Anthony Fauci as director.
How much money was that?
The amount of the budget that Fauci directly controlled was $6.5 billion annually, but the amount that he controlled through his authority not only over his agency's budget, the NIAID budget, but the budgets of all U.S. entities engaged in biodefense research would double or triple that number.
And it seemed from what I read that he had not only no qualms or serious concerns about this research, he was a huge proponent for it. And he didn't seem like he thought anything bad was going to come out of it. Is that your impression?
His appointment as the authority for biodefense research across U.S. agencies was contingent on his pursuing the research aggressively up to and across red lines. That is precisely why he was assigned that position. When he was assigned that position, a special arrangement was made in which his salary was more than doubled, making him at that point and through to his retirement the most highly compensated member of the United States government, with a salary substantially higher than that of the President of the United States, substantially higher than that of any justice on the Supreme Court, a salary higher even than the director of the National Institutes of Health, who was nominally his supervisor, his direct report, the person to whom he reported in the chain of command. Again, the position and the salary and the influence that came with that position were contingent on approaching and crossing red lines. Prior to that, he was the director of one of 27 institutes and centers at National Institutes of Health. Most could not name the director of a single institute or center, including researchers funded by those centers. After the reassignment as head of biodefense, he had a direct line to the President of the United States in each administration from that point onward. He had a representative from the Office of the Vice President placed as a political commissar in his offices, in NIAID offices, to oversee and ensure that the research funds were being used in the way that the Vice President, in this case Vice President Cheney, intended. So the position, the salary, the influence, and the visibility and impact all were contingent on approaching and crossing red lines. When administrations changed, next had the Obama administration, the position continued, and that position continued, and all of the accompaniments of that position in terms of salary, influence, and impact continued up until his retirement at the end of 2022.
So you organized a petition. I think this was in 2005, 4 or 5, am I correct?
2004, 2000, I'm remembering it's 2004.
Okay, you organized a petition signed by 758 scientists, including 2 Nobel Prize winners, to protest this shift in tens of millions of dollars in federal research and taking it away from pathogens that you and I probably would consider a major health concern into this other kind of bioweapons research. What was the NIH response? And also, if you could elaborate on that petition, what you wanted to come of that, and then what was the response from NIH?
I'll first comment on the background of the open letter. So the open letter followed a period that started in 2002 and continued to 2004, in which Fauci announced that research funded by NIAID would be reprioritized. And the approximately half of the NIAID budget, or approximately two-thirds of the NIAID budget that covered research other than HIV/AIDS, previously was directed to a range of pathogens that cause disease in humans. Bacterial pathogens, fungal pathogens, viral pathogens, protozoal pathogens. An announcement was made, communicated to researchers receiving NIAID funding, that henceforth there would be priority pathogens. categorized as Class A, Class B, and Class C NIAID priority pathogens. The prioritized pathogens were pathogens that were drawn from the list of pathogens that were deemed to have impact and potential for use as biological weapons agents. That is to say, pathogens that had been used in the past as biological weapons agents were perceived as being likely to be used in the future as biological weapons agents.
What would people recognize as a few of those?
And once you start with the one that prompted the entire reprioritization, and that was anthrax.
Mm-hmm.
So from bacteria, you would think of the anthrax bacterium, the plague bacterium, and a handful of others. Among viruses, you would think of the 1918 pandemic influenza virus, highly pathogenic avian influenza viruses like avian influenza virus H5N1. One would think of the smallpox virus. One would think of other orthopox viruses, such as the mpox or monkeypox virus. One would include the Ebola virus, the Marburg virus, the Lassa fever virus. And at that point, with relatively limited visibility until 2002 and 2003, coronaviruses. In 2002 and 2003, there was an outbreak of the original SARS coronavirus, SARS-CoV-1, and the impact of that outbreak, which was fortunately rapidly contained, indicated that SARS viruses should be also considered as biological weapons agents. So that would be the group of pathogens that people might recognize the names of. And so these were categorized based on their level of threat as Category A, Category B, or Category C NIAID priority pathogens. And funds were assigned for research on these. And a strong message was communicated to NIAID-funded researchers, both on the NIH campus in Bethesda, Maryland, and across the United States at universities, that you should switch from studies of pathogens that you have been studying in the past to studies of Category A preferably, Category B less preferably, or Category C less preferably pathogens. These would be funded. It would be easy to get support. Funding for other categories of pathogens would wither. The open letter then was a campaign by researchers who were funded by the National Institutes of Health and or who had reviewed funding proposals by the National Institutes of Health for research on bacterial pathogens. So category of pathogens, NIAID-funded research. Every person who signed is a person who was awarded a grant and or who had reviewed grant proposals to determine who would be awarded a grant. So this was not just a set of 750 scientists. These were the 750 scientists who were engaged in this research. And virtually all scientists who had received NIH support for this category of research or reviewed proposals for this category of research signed to say that the reprioritization was a grave mistake. It was a grave mistake primarily from the standpoint of the scientists in that it was removing support from urgently needed areas of research with direct public health research and relevance, and instead directing it to a class of research with, at most, a small connection to public health and public health relevance, and with serious concerns in terms of potential accidental or deliberate release. So the letter was submitted to the then NIH director, Elias Zerhouni, and of course to the then NIAID director, Anthony Fauci. The response from NIH and NIAID was to dismiss the letter, initially to deny that there had been any change of funding. This was simply a denial that could not be accepted. The people signing the letter are the persons who had seen their funding lines evaporate unless they switched their research from non-bioweapons agents research to bioweapons agents research. They were not going to be persuaded. But initially the response was a denial. Subsequently, the response was an acknowledgement Anthony Fauci organized a meeting in Bethesda, which brought many of the signatories to express their concerns. He attended only the first minutes of that meeting, and the meeting resulted in a report, but it did not result in changes.
Were you there at that meeting?
Yes, I was. And the response to the open letter marked the beginning of what we then saw starting in 2004 and continuing through to Anthony Fauci's retirement at the end of 2022, of a response by doubling down on previously made decisions. So when concerns were pointed out, concerns that in essence represented a vote of no confidence from his entire or nearly his entire portfolio of researchers performing research on bacterial infectious diseases, the response was to double down. I have made the right decision. This decision has brought the NIH additional funding that had previously been going to other areas, such as the Department of Defense. It's available to you. You should be happy to take advantage of it. And all you have to do is switch to start working on a Category A, Category B, or Category C pathogen. The same kind of response then occurred when incidents in that research on Category A, B, and C pathogens occurred. The first significant incident was the reconstruction of the 1918 pandemic influenza virus. So this is a virus that had last been present on the planet shortly after World War I. At that point, it infected nearly every person on the planet and killed approximately 1%. So an extremely lethal, extremely concerning pathogen, which had disappeared from the scene, and as a result of which the population had, particularly anyone who was under the age of 60, had very limited immune protection against that pathogen.
Could you please explain how that pathogen came to be wiped off the face of the earth?
This is what occurs. You had a pathogen that you had a pathogen that caused a global pandemic, infecting nearly every person on the planet and killing 1%. Those who were not killed were now immune. So when it appeared on the scene, there was not pre-existing immunity to a virus that would present two key surface proteins, the two key immunogens of an influenza virus, the H or hemagglutinin protein and the N or neuraminidase protein. These were new types, H1 and N1, not recognized by the immune systems of people who were present. People became infected. They suffered very severe symptoms. But those who were infected and survived now had immunity to H1N1 viruses. And H1N1 viruses continued to cause infections, but the infections would be contained and would have more limited symptoms. And this pandemic virus then subsides to become a seasonal influenza virus, and in time was superseded by other seasonal influenza viruses, left the scene. And again, the population, as we're now in the 21st century, in 2004, the population had essentially no pre-existing immunity to H1N1 influenza viruses because they had not been exposed to viruses of that type for decades. So researchers obtained sequence information, the full genome sequence of the virus, by exhuming corpses from the Arctic tundra of persons who had been killed in the 1918 pandemic, extracting nucleic acids from the corpses and reconstructing the sequence of the virus. And this was an important accomplishment in science, to be able to recreate or deduce the actual sequence of the pathogen that had caused the pandemic. The researchers took a next step, which was much more concerning, and that next step was to reconstruct the 1918 influenza virus, constructing fully infectious virus using the sequence and newly developed synthetic tools that enable one to synthesize a full viral genome, introduce that into cells, and obtain fully infectious viral particles.
How many
Uh,
people in the world do you think can do this kind of experiment?
so it depends on the pathogen and it depends on the point in time when we're asking. That was in 2004 for influenza virus. Influenza virus has a relatively short genome, about 8,000 base pairs, and in segments that are relatively easy to synthesize, even with the technology of that time, there were perhaps a dozen who could have done that in 2004. Today, that could be done in hundreds of laboratories worldwide
Mm.
and would be considered relatively simple. So, but in any event, the 1918 influenza virus was reconstructed. That made front page coverage in the New York Times, in the Washington Post, all three networks. And the most frequent expression was an expression of concern. Why
Yep.
was this research being done? How was it performed without risk-benefit assessment? And it became very clear very early that the research had been funded by the NIH, by Anthony Fauci, and had been funded without having performed any form of risk-benefit assessment. There was pushback from Congress as to the question why this research could have been and would have been funded without a risk-benefit assessment. And Fauci responded the same way he had responded, I believe, a year before to the open letter, which was to double down and to state that this research was of such crucial importance for U.S. public health and for U.S. national security that to not have done it or to have paused doing it while carrying out a risk-benefit assessment would have been completely unacceptable.
What did he claim the benefits were?
The benefits he claimed were the same claim that would be made in any similar circumstances. The standard claim would be that the knowledge gained by reconstructing the virus and studying its properties will help us prevent pandemics in the future and help us respond to pandemics in the future.
But that's not correct.
No part of that is correct. The information that came from the reconstruction and characterization of the virus provided fundamental scientific information, but it provided no applications. It did not enable the development of a new vaccine. It did not enable the development of a new therapeutic agent. It did not enable prediction or prevention of pandemics. And there was no possibility, no plausible route by which the information would lead to any of those benefits, even from the start. And thus, if a risk-benefit assessment had been performed,
yeah.
And in a true risk-benefit assessment, one first enumerates the anticipated risks and the enumerated benefits. The enumerated risk would be an accidental or deliberate release of the virus. A lab accident, such as a lab infection of a lab staffer, followed by transmission to the general public, would spark another pandemic and could kill millions. A deliberate release like the 2001 anthrax mailings could do exactly the same thing. Another area of risk would be that an adversary, a rogue nation, or a sub-state adversary like a terrorist organization would use the information to reconstruct the virus and deliberately release the virus. So there were concerns about the material, the virus itself being accidentally or deliberately released, and concerns about the information, the sequence of the virus and the method of reconstruction, prompting a release by an adversary nation or sub-state organization. Well, the benefits would be asserted that this would provide information that would inform vaccine development, drug development, or prevention of pandemics, but there would be no route from that claim benefit to how to obtain that claim benefit. And so we would have a series of existential risks, truly existential level risks, balanced by an ostensible set of claim benefits, but claim benefits that cannot be actualized, had no route to actualization. And we can now look back with the benefit of hindsight and say, was the information from the 1918 influenza virus reconstruction informative, and was it useful, and did it have applications? And so it was informative. It provided information that is information that resulted in scientific papers with new findings, but it had no practical benefits, none whatsoever. And that would have been foreseeable had a risk-benefit assessment been made, and had a risk-benefit assessment been made, surely, and independent and objective set of observers would have concluded that the risks outweighed the potential benefits and that the research should not have been performed. But Fauci's response at the time was, I did the right thing. I would do it again. And the research prioritization of this category of research continued. Again, the 1918 influenza virus was a category A NIAID priority pathogen, continued to be funded, continued to be prioritized. The avian influenza virus H5N1 was another. The next incident on the timeline occurred in 2011. So in 2011, two research laboratories, both funded by the NIH, both funded from NIAID, Anthony Fauci's units, performed research that enhanced the likely transmissibility and pathogenicity of avian influenza virus H5N1. So two laboratories, one in the United States at the University of Wisconsin-Madison, and one outside the United States, but still funded by the United States, by Anthony Fauci. That second one in the Netherlands at Erasmus University. Started with avian influenza virus H5N1. This is the bird flu that you read about in the papers daily as infecting large numbers of animals in the United States and elsewhere and posing a high risk of pandemic breakthrough.
And what's the mortality rate in humans for this disease?
As of 2011, it was believed that the case fatality rate was approximately 60 to 70%, which is to say that between 60 and 70% of infected persons died. But this was a virus that did not transmit efficiently from human to human or from mammal to mammal by an aerosol respiratory droplet route. So it's a virus that was concerning in terms of its expected case fatality rate, potentially as high as 60 to 70%, but limited in its ability to transmit. The two research groups, again, both with NIH funding, sought to engineer versions of avian influenza virus H5N1 that could transmit efficiently from mammal to mammal through an aerosol respiratory droplet route. And both groups accomplished this. Both groups accomplished this, identifying genetically modified viruses that now had the ability to transmit from mammal to mammal through an aerosol respiratory droplet route. Again, this resulted in front page coverage in the New York Times and in the Washington Post and on all four networks. Again, this resulted in an outcry, with the most common response being that this is research that should not have been performed. And when it was learned that the funding had been approved without a risk-benefit assessment, there was real pushback from both houses of Congress, with senior members of Congress in both parties saying, why was this research funded? We were told that changes would be made to ensure that risk-benefit assessment would be performed. But now we have something that is even more threatening than the 1918 influenza virus reconstruction. And again, we find it was funded without a risk-benefit assessment. Mm-hmm. And the response was the same. The response from Anthony Fauci was to double down and say this research was so urgently necessary for U.S. public health, U.S. national security, that to have not done it would have been the mistake, or to have delayed it while performing a risk-benefit assessment. That would have been the mistake. Fauci was under significant pressure from senior members of Congress in both parties who were concerned about this, and his position was indeed threatened at that time. He was able, however, to recruit the then new NIH director, Francis Collins, to write an opinion piece, an op-ed for the Washington Post entitled "A Flu Risk Worth Taking," stating that this research was so crucially important for public health and national security that it was a risk that needed to have been taken, and that Fauci and by association, Collins, had taken the right decision, had made the right decision by proceeding with the research and funding it and not pausing for a risk-benefit assessment.
Well, I read that article many times, and I didn't come away with any understanding of why he thought that the risk was worth taking. Do you understand why he thought it was worth it.
Again, the same ostensible benefits would be cited, that the research would result in information that would provide new vaccines, new drugs, means of predicting and preventing pandemics.
Again, though, we should just, I was just, but not true. Did he know? I mean, I know we can't know what anybody really thinks, but do you believe he knew at the time that was that their true intention? I guess is what I'm asking you.
That was always an ostensible benefit, an ostensible set of benefits, an ostensible set of justification, not the actual motivation for the funding agency to fund the research, not the motivation for the researchers to perform the research. And certainly there has been no interest in using that research to develop a new vaccine or a new drug. It simply has not happened.
So just to be...
The research has never been used for, has never contributed to the development of any vaccine or therapeutic.
So you said it wasn't the ostensible reason. And then there was another, more real reason behind that. Can you just lay it out? What was that reason, really?
The high pathogenicity avian influenza viruses were a categorized priority pathogen for NIAID, which is to say that they are viruses that are on a list of known or anticipated biological weapons agents, and research on those agents was prioritized, starting, as I said, after the 9/11 terror attacks and the anthrax mailings, as part of the large change in funding and the large change in federal oversight of U.S. biodefense efforts. This was an example of research that would not even have been contemplated, much less performed, before 2002, even if all of the experimental technologies required existed and had been in place.
Okay.
It would not have been performed, it would not have been anticipated until and unless the research that was being performed on public health relevant infectious disease biology was deprioritized and research on biological weapons agents was prioritized. Researchers, again, were compelled, if they wished to remain funded, to transition their research from public health relevant research to biological weapons agents relevant research. And then they had to struggle to conceive of experiments to do in order to propose proposals on biological weapons agents. And it became clear early on that the most successful proposals would be proposals that used fully infectious biological weapons agents and performed enhancements and threat characterization of enhanced agents. And this is an example of that category of research. That is the reason this category of research was performed. Mm-hmm. From the standpoint of the researchers, there were key incentives. The first incentive is that research to enhance a pathogen is straightforward. It doesn't require an original hypothesis. It doesn't require sophisticated methods. It doesn't require sophisticated instrumentation. And it very frequently is successful. So easy experiments. Priority one. Incentive one. Prioritize funding. This is research that was, because of the prioritization, virtually insured funding. Funding is a major incentive. Incentive two. And prioritize publication. Researchers learned that research, particularly if it was transgressive, particularly if it was so transgressive that it would get general media coverage, like the 1918 influenza virus reconstruction, or like the H5N1 mammalian transmissibility experiments, if it was transgressive enough to get on the front page of the New York Times or the front page of the Washington Post, that research would be sought out by the most important scientific journals, the highest impact scientific journals, because the editors at those journals are seeking visibility and impact, particularly rare cases of visibility and impact that make a crossover to the general media. So third incentive, easy publishability. So from the standpoint of the researchers, this research was easy to do, easy to fund, and easy to publish that is a very strong incentive structure. From the standpoint of the funding agencies, a decision had been made to prioritize biological weapons agents research. And a decision had been made specifically to prioritize research that approached, reached, and crossed those red lines set by the BWC.
So the public is often told that this research is done because they're afraid about a spillover. They're afraid that H5N1 is going to evolve to be transmissible human to human because it's in animals.
That is a concern that we are facing currently as this virus, a decade or more than a decade after those experiments, now has moved partly toward reaching the point where it might break through to humans. But the point is, there are multiple ways for a virus to evolve, almost an infinity of ways, and there are multiple routes that could be pursued. The route that was identified by the research lab at University of Wisconsin-Madison, the route that was identified by the university in the Netherlands at Erasmus University, those routes are not the routes that likely would occur if this virus makes a pandemic crossover in nature.
Right.
And so the information provided by those studies defined ways the virus could approach humans. It could pose higher pandemic potential, ways the virus could achieve higher pandemic potential, but not the way that will occur next, not the way that necessarily will occur in this decade or this century or this millennium. As a result, you can't use it to develop a vaccine or a therapeutic because there are so many possible ways the virus might achieve pandemic potential. No company would ever in advance invest funds to address one way when there are 10,000 alternative ways. The probability that the correct vaccine or correct therapeutic would have been developed is essentially nil.
How concerned are you right now about what's happening, and why is this being contained in animals?
I am concerned, and I'm concerned about the lack of attention that the current administration has assigned to this threat. But the experiments that were performed by the University of Wisconsin-Madison lab and the Erasmus University lab don't inform us in any way about the risk, don't enable us in any way to address the risk, to predict or prevent the risk. If they had not been performed, or if they had been performed, we would be in exactly the same position today. So their only consequence was to develop not 10 years or 100 years or 1,000 years from now, but a decade ago, more than a decade ago, a previously non-existing, non-natural threat, which has been in a freezer and which at any point through an accident or deliberate action could leave that freezer and create pandemic potential instantly.
How does H5N1 spread between humans?
At this point in time, it transmits extremely poorly human to human, and it is even controversial whether it transmits at all human to human.
Mm-hmm. There's concern about drinking milk. There's concern about eggs. There's concern about eating chicken.
Mm-hmm.
I'm seeing all kinds of articles and all kinds of information and potentially misinformation. And I just don't know how concerning those things really are.
It's a concern, but at the moment, I don't think it's an immediate concern. But a snap of the fingers, and it could be. So it's something that should be monitored more closely than it is now, and steps should be being taken that are more intensive by far than the steps that are being taken now to monitor this virus in livestock and in humans, and to contain this virus in livestock and in humans. But relative to our discussion, the important point is that the research that was performed in 2011 and 2012, as well as all the follow-up research on those engineered viruses, has contributed nothing to our understanding of the risk we face today, and absolutely nothing to responding to that risk we face today. So it was creating a risk that fortunately has not yet materialized of accidental or deliberate release, but which could at any point.
Are lab leaks common?
Lab leaks are extremely common. So the term it would be better to refer to laboratory acquired infections and laboratory releases. And so laboratory acquired infections and laboratory releases are common. Laboratory acquired infections are especially common. Researchers, particularly if they're working with a pathogen that is transmissible through aerosol route, frequently become infected with pathogens under study. In many cases, those infections aren't recognized. If an infection isn't recognized, if it's asymptomatic or oligosymptomatic or even symptomatic but not recognized, the researcher then has the prospect of infecting others in the community, which can give rise to a larger outbreak. In the United States, there is a requirement to report breaches of containment, breaches of biocontainment, and laboratory acquired infections for a subset of pathogens that are deemed to have highest risk for use as biological weapons agents. A subset of about 60 pathogens and biological toxins referred to as select agents. And it includes ones that we mentioned, like the smallpox virus, like the 1918 influenza virus, like the avian influenza virus H5N1, like the SARS virus, like the anthrax bacterium and the plague bacterium. For that subset of pathogens, and only for that subset of pathogens, there is a requirement to file with the federal government a theft loss release document, a TLR, for any instance of theft, loss, or release of the pathogen. And the United States government releases summary reporting information on those filings biannually. So every 2 years, a summary is released of the number of reports received. And over the entire period in which the TLR filings have been received and monitored, there has been an average of between 2 and 4 filings each week. So this is in the United States alone for a small subset of pathogens, the subset of highest risk for use as a biological weapon agent, a breach of containment, a loss, or a theft occurring 2 to 4 times each week on average over the entire period of the biodefense expansion from 2003 to the present.
Yep.
Canada maintains a similar system of reports and has a similar number of 2 to 4 per week. The United Kingdom has a similar system of reports, very similar number, about 1 to 2 per week. These are not common. These are occurring at a shockingly high rate. And a significant fraction of the TLR reports, the theft loss release reports, are laboratory acquired infections. That's the highest concern breach of biocontainment, because that's the one that if a researcher is infected and doesn't recognize the infection and leaves the lab, and if the infection is communicable, it can be communicated to others.
Has that happened before in any kind of major way? And
Well,
maybe
that
you
has
could make
happened
you could make the argument for COVID, but I'm wondering what you think prior to COVID.
The original SARS virus, SARS-CoV-1, caused lab-acquired infections in Singapore at biosafety level 3, twice in Beijing at biosafety level 3, and in Taipei at biosafety level 4. And the incidents in Beijing not only involved laboratory-acquired infection of laboratory staffers, but also transmission from the infected staffers to the community, with an infection of at least one healthcare worker, an infection of at least one family member, and with the death of a family member. SARS-CoV-2, the virus that causes COVID-19, also has caused documented laboratory-acquired infections. There was a lab-acquired infection in Beijing in 2020 at biosafety level 3, and in Taipei, Taiwan in 2021 at biosafety level 3.
It's really shocking to hear how many times this has happened. I actually didn't have a sense of the scope of these reports, and these things are happening in our backyards.
Yes, and it's important to note that the numbers, the frequency of laboratory acquired infections and other biocontainment breaches in the reports in the US and in Canada and the United Kingdom are effectively the same. This is something that is not an issue occurring only in certain laboratories in certain countries. This is the norm.
I should mention that you co-founded Biosafety Now with Bryce Nichols, which I am on the advisory board, which has been really interesting and fascinating education for me. Listening to all of you talk about these really, really incredibly important issues, and it's really fired me up, as you know, to try to understand them better and communicate them better to the public, which is why talking to you today is so important to me, and just going to really open people's eyes to the reality of this happening right in their backyards. What was the mission for you when you first started to discuss this with Bryce and put Biosafety Now together? What was your goal?
So starting with the United States, in the United States, there currently is almost no national level review of biosafety and almost no national level review of biorisk management. That is to say, risk benefit assessment of the highest risk category of biological experiments.
Yeah.
For biosafety, for example, in the United States, there are no rules with force of law, no enforceable laws, no enforceable regulations for biosafety of any pathogen other than select agents, the approximately 60 agents that are deemed to have highest potential for use as biological weapons. And even for the select agents, the only rule with force of law, the only enforceable rule, is that the laboratory must have a biosafety plan and must approve that plan annually. The content of the plan is entirely at the discretion of the researchers and their institutions. The only requirement is that a written plan must exist. Mm-hmm. That is biosafety in the US. Biorisk management in the United States is similarly limited. There are no regulation or rules with force of law at all for any pathogen other than the smallpox virus that pertain to risks. There is a policy, an advisory policy, not an enforceable rule with force of law, but a policy that requires risk benefit assessment at the federal level by a committee established by the Secretary of Health and Human Services for research that is reasonably anticipated to increase either the transmissibility or the virulence of a potential pandemic pathogen. But that policy has been largely ignored. It exists on paper, but it has existed in practice almost not at all. The majority, indeed the overwhelming majority of projects that require that risk-benefit assessment under the policy never received it. For example, the project in Wuhan that likely gave rise to the COVID-19 pandemic was funded without the risk-benefit review that's required by that policy. And the policy has been non-operational because funding agency officials simply have refused to forward projects to the risk-benefit review committee for review. So Francis Collins at NIH, Anthony Fauci at NIAID simply chose not to flag projects for review, not to submit them for review, and no review occurred. So in this context where there is almost no regulation with force of law for biosafety and no regulation with force of law for biorisk management for any pathogen other than smallpox virus, Bryce Nichols and I felt that it was urgent, starting with the United States, to change that. Scientists work with a large array of regulations. There are regulations with force of law, enforceable regulations for research with vertebrate animals, and for research with human subjects, and for research with chemical carcinogens, and for research with hazardous chemicals, and for disposal of chemical waste, and for research with radioisotopes, but not for this category of research, which poses truly existential risk that dwarfs any of those categories of research I've just mentioned. So we felt that it was important to found an organization that would communicate to the public, to the press, and to policymakers the actual status of current U.S. government oversight, which is essentially none, the importance of enhanced oversight, and then that would push for enhanced oversight. And in particular, that would provide a means for the public to be considered a shareholder and for the public to participate in the evaluations of risks and benefits and to participate in influencing discussions by policymakers and decisions by policymakers.
Can you comment on the group of scientists who vehemently oppose regulation?
It's a fairly small group of researchers with the strongest concerns coming from those researchers who in 2003 and 2004, when the reprioritization of NIH funding was occurred, enthusiastically made the jump from public health research to biological weapons agents research. And when they made that jump, not merely moved to change the pathogens that they studied, but also to change the approaches to focus on transgressive research, research that would increase the risk posed by the pathogen by making it more transmissible or more pathogenic or more able to overcome a vaccine or therapeutic, or to reconstruct a previously eradicated or extinct pathogen. So the highest risk category of experiments. That small group of individuals have been richly rewarded. They've been richly rewarded by opportunistically making the leap to a different area of research and a different kind of research and pursuing it aggressively. Any form of risk-benefit oversight will result in a restriction of their freedom to operate. Most likely will result in a restriction of their funding and their publishability. And the key incentives I discussed before in terms of easy experiments, easy fundability, and easy publishability would come to an end. They have fought this tooth and nail. Until the end of 2022, they have been assisted in this fight and led and directed in this fight by Anthony Fauci and Francis Collins, the NIAID and the former NIAID director and former NIH director. One of the motivations for founding Biosafety Now was the realization that with those individuals no longer controlling the levers of funding, no longer in the case of Fauci having a $6.5 billion budget with which to win friends and influence people. In the case of Francis Collins, no longer having a $35 billion budget with which to win friends and influence people, that there would be a possibility for the first time to have a broad impact with involvement of the public and with concerned scientists and concerned science policy specialists to try to enhance oversight in the U.S. and to basically return to the situation before 2003, to walk back all of the changes that were made in the prioritization of biological weapons agents research, all of the changes that were made under Anthony Fauci's direction with the guidance and overall direction from Dick Cheney in 2003, 2004, 2005, and the time since.
It is kind of amazing. It's an amazing moment for Biosafety Now in the advent of Dr. Jay Bhattacharya and Marty Makary.
As you know, Jay Bhattacharya is a member of the board of directors. Marty Makary is a member of the board of advisors now. So two of our board members now will have key positions of influence in the coming administration, assuming they are confirmed, and will be in a position to advance objectives of Biosafety Now. That was also part of the motivation for founding Biosafety Now, was the recognition that the United States government was on a knife edge balance with nearly equal partisan balance. And that meant there was a situation where public influence could have real impact, and public influence that would change the vote of just one senator or just one representative in a House of Congress where the balance is decided by one, two, three, or four, or five could make the difference. And so that was a unique opportunity for the public, for the first time, scientists who've been trying to have impact for 2 decades, and science policy specialists who've been trying to have impact for 2 decades, to potentially have real impact. And so I agreed with Bryce that it was important to form an organization that could bring together the public, scientists, and science policy specialists and apply pressure, select points of pressure at select points within the political system that would have greatest impact. And we now stand with 2 members of our boards in positions, likely to be in positions of influence in the administration, with both houses of Congress in the 119th Congress, having majority control from a party that has committed to pursue these issues, committee chairmanships in both the House and the Senate moving into the hands of individuals who've committed to pursue these issues. And so this is a real sea change.
Yeah, absolutely. It's been incredible to watch this evolution because I never thought it was going to happen. And I have worried that this was the biggest threat of our time that we're facing now. And it's really hard for me as a member of the public to have these conversations with people, with friends and family, and have them take it seriously because it sounds so outrageous the first time you hear about it. And so again, these conversations.
It's just remarkable that people say, don't they have institutional review boards that monitor this?
Yeah.
People are familiar with the regulations that exist for human subject research.
Yes.
Some are also familiar with the regulations that exist for vertebrate animals research. And it's incomprehensible that research on a human subject of any kind, even the least intrusive, requires an intensive prior review and approval process, whereas research to take a biological weapons agent and maximize its ability to cause harm requires no review whatsoever. Or if it requires review, that review is ignored by the funding agency officials who simply nullify the requirement for a review by ignoring it.
Yeah, it's stunning. My parents watched my interview with Dr. Redfield recently, and I haven't really tried to engage them in this topic. And it's so overwhelming, and where do you even start? And they watched this interview with a lot of interest. And my dad called me, goes, "Shouldn't people go to jail for this? This is the craziest thing I've ever heard in my life." And I said, I agree with you, Dad. You know, and that is the right reaction. Like, to me, that's the appropriate. He was outraged. He was shocked.
Well, I think that's the reaction of any informed person who does not have a vested interest in continuing the status quo. Next point on the timeline would be 2014. So in 2014, a series of embarrassments occurred within the U.S. biodefense program, the bioweapons agents research programs in the United States at premier federal government laboratories. One of the incidents occurred at the CDC laboratory in Georgia, a CDC laboratory in Georgia, and involved exposure of dozens of CDC staffers to fully infectious, fully virulent anthrax spores inadvertently, requiring prophylaxis, antibiotic prophylaxis of all the exposed individuals, fortunately not causing infections. A second one occurred at a CDC facility, again in Georgia, and involved a researcher who, through an interchange or cross-contamination of samples, accidentally sent avian influenza virus H5N1 to another laboratory. The other laboratory was expecting to receive a low pathogenicity influenza virus, received avian influenza virus H5N1, and was startled to find that their experimental animals all were dying. And so this was an accidental interchange or cross-contamination of materials, followed by a transmission through the mail to another laboratory, where only rapid observation, and rapid action by that laboratory detected the problem and allowed it to be corrected before broader consequences. A third incident occurring in 2014 was finding a broken glass tube labeled smallpox in a laboratory operated by the FDA and the NIH in the Washington, D.C. area. And in very short order, 3 such embarrassments all appearing, there were responses at multiple levels. One of those responses was the formation of a group of concerned scientists and science policy specialists. The group was called the Cambridge Working Group, that issued a declaration that there was an urgent need to address the biosafety, biosecurity, and biorisk management shortcomings that incidents like the 1918 influenza virus reconstruction, like the avian H5N1 engineering, and like these 3 events occurring in 2014 were underscoring. Second was a congressional hearing in the House on the biosafety incidents, with particular focus on the CDC anthrax incident, and reaching the conclusion that there was an urgent need for enhanced oversight. These calls for enhanced oversight were fiercely resisted by Anthony Fauci, who coalesced a group of scientists and science policy specialists who formed a counterbalancing organization called Scientists for Science that argued that any calls for oversight would reflect the same kinds of errors that the Pope made in asking Galileo to recant his view of a heliocentric universe, and that a call for oversight of biosafety is the same as the Pope and the Inquisition addressing Galileo.
Wow.
The Obama administration stepped in through the Obama Office of Science and Technology Policy in October 2014 by imposing a federal government-wide pause on funding of research that would be reasonably anticipated to increase either the transmissibility or the pathogenicity of an influenza virus, a SARS virus, or a MERS virus, with those 3 being recognized as the most concerning potential pandemic pathogens that would be easy to genetically engineer with the techniques that were available as of 2014. The policy called for 2 things. It called for a pause on federal funding, and it calls for a deliberative process that would identify a framework of risk-benefit assessment that would be put in place and would be mandatory and enforceable. And there would be no lifting of the pause until such a framework were implemented. Okay. The funding agencies were asked to review their portfolios, identify any funded research that met the definition. They did not do so. So Francis Collins and Anthony Fauci developed only a partial list, and they developed a list of 18 funded projects. Those 18 funded projects received letters informing them of the funding pause. But within 2 months, for at least 7 of the 18 projects, Anthony Fauci and Francis Collins lifted the pause. And they lifted the pause by taking advantage of a provision that stated that a funding agency official, if the funding agency official declared the research to be of urgent necessity for public health security and national security, could lift the pause. So 18 projects were paused. Within 2 months, 7 had their pause lifted. It is thought that others of the 18 also had their pause lifted subsequently. But even more fundamentally, not all projects that met the definition of the pause were paused. In particular, the research on genetically engineered SARS viruses being funded by the NIH in Wuhan was not identified as subject to the pause, even though during the period of the pause, NIH was informed in writing that research was being performed as part of that project that clearly met the definition of having reasonable likelihood of increasing transmissibility or pathogenicity of a SARS virus or a MERS virus. The researchers were performing engineering on both SARS viruses and MERS viruses that would be expected to have that consequence. So that policy then remained in effect until the beginning of 2018. At the beginning of 2018, the deliberative process came to its end, so the pause ended, and a new policy framework was announced. That policy was known as the Potential Pandemic Pathogen Care and Oversight Policy, or P3CO for short, and it called for a mandatory HHS secretary level risk benefit assessment for any research reasonably anticipated to increase the transmissibility or virulence of a potential pandemic pathogen. So it is a policy that uses a definition that matches the definition in the pause, research reasonably anticipated to increase transmissibility or pathogenicity or virulence, and expands to the full set of potential pandemic pathogens, not just influenza viruses, SARS viruses, and MERS viruses, but those viruses and also other potential pandemic pathogens. That policy came into effect in 2018. Two of the projects that still had been paused out of the original 18, so two of the projects were grandfathered into the new policy to receive risk benefit assessment and receive that risk benefit assessment early in 2018. But after that point, Fauci and Collins only submitted one additional project for review. Out of the estimated 1 to 3 dozen projects that would meet the definition that they funded each year, they submitted only one in the entire period of 2018, 2019, 2020, 2021, 2022, the entire time up to their retirement. So in that 5-year period, out of 1 to 3 dozen per year, that's 60 to 180 projects, they submitted one.
Wow.
Most importantly, they did not submit the project on engineering of SARS viruses occurring in Wuhan. Even though they had been informed in writing, or at least Anthony Fauci had been informed in writing. So they did not include the virus on engineering of SARS viruses for increased transmissibility and pathogenicity in Wuhan, even though the NIH had been informed that that project, as of 2018, not only was reasonably anticipated to increase transmissibility and virulence, but had blown through all possible standards and trigger points for coverage. NIH was informed that the researchers in Wuhan, as of 2018, had engineered SARS viruses to have a 10,000 times increase in viral growth in lung, a 1 million times increase in viral growth in brain, and a 3 times increase in lethality when tested in mice that were engineered to possess human receptors for SARS viruses, so-called humanized mice. The NIH was informed of that twice in 2018, and that information should immediately have triggered a suspension of funding, followed by risk-benefit review before resumption of funding. Fauci chose not to flag the research, not to suspend funding, and not to forward the proposal for review. That is the research project that, by all indications, caused COVID-19, killing 20 million and costing 25 trillion. That project could have been prevented had, at any point from 2002 to 2019, Anthony Fauci listened to concerns that were expressed from the public, from scientists, and from science policy specialists, and from the public's representatives in Congress, and hesitated, or introduced risk-benefit review, or even complied with the minimal terms of the policies that were in effect. but he chose at each time to double down, to violate the policies, knowingly and willfully violate the policies. Those violations, it appears, are responsible for 20 million deaths and $25 trillion in losses.
What has the role, since all of this has happened, of NIH been in trying to hide their culpability?
Well, when it became apparent that SARS-like pneumonias in Wuhan were caused by a SARS-like coronavirus that was a new coronavirus, all informed persons, without exception, understood that the probability was this was a laboratory release. All informed persons understood this precisely because this had been under discussion by science and science policy specialists for the entire period we're discussing. In 2015, this specific project, a project being carried out jointly by the University of North Carolina at Chapel Hill and Wuhan Institute of Virology, this specific project, the joint project of those two institutions, engineering SARS viruses by replacing spike genes of one SARS virus with spike genes of another, creating novel viruses with potentially increased transmissibility and potentially increased virulence. This project was singled out as the project of all projects being carried out in the world most likely to cause a pandemic. It was singled out and was the subject of an entire afternoon session, a half-day session at a meeting that was jointly sponsored by the Royal Society of the United Kingdom and the National Academies of Science, Engineering, and Medicine of the United States, a meeting held in UK at which the project was discussed at length. Everyone in that room, everyone in the broader discussion among scientists and science policy specialists knew this project was being carried out, knew it was being carried out in Wuhan. And when they heard that a new SARS virus, different from the original SARS coronavirus, but closely related to it, was responsible for SARS-like pneumonias emerging in Wuhan at the start of January, every person involved, without exception, knew that lab origin was a very real possibility.
Yes.
A very real probability.
I can tell you from my point of view, writing a book, finishing the book at that time, when SARS-CoV-2 broke out, our publisher put the book on hold until we could write a COVID chapter, because we were writing about a whole other group of pathogens, tick-borne and vector-borne diseases. And so we did lots of interviews with people we didn't know at that time, but they were involved in this world. And I will just say, and many of those conversations were off the record, and it was really more informational background stuff for us to understand better what was going on. 100% of them said that this was bioengineered.
In January and February of 2020, it was not clear yet whether this would be a containable outbreak like the original SARS-CoV-1 outbreak in 2002, 2003, or whether this would become a broader pandemic. But it was clear to all informed persons, without exception, that there was a very high probability, very real probability this had a lab origin. And it was clear, absolutely clear, to the funders of this research, including Anthony Fauci and Francis Collins, that they funded research that surely had caused an outbreak and potentially might cause a pandemic. Their response to this was to recognize their culpability, but prevent the public and the press and policymakers from recognizing their culpability. They moved into cover-up mode in which all tools at their disposal, the large budgets at their disposal, the $6.5 billion budget in Anthony Fauci's unit, the more than $30 billion budget in Francis Collins' unit, those budgets, their staff, their credibility, and the credibility of the persons who perform research with funding from those budgets were deployed to prevent the public, the press, and policymakers from learning the facts.
You just gave some context, but I think people that haven't have only been reading headlines for the last 5 years, called people like you or like me conspiracy theorists. There was a major smear campaign, and it even like me got to me. It wasn't just people like you in a position of authority in this area. I would say something, and it was really, really serious pushback. And I thought it was so coordinated and so shocking. And I thought that it was so virulent. What has it been like for you to have been in the crossfires of this and been in the press and had hit pieces written on you in the LA Times? I mean, crazy stuff that's just 100% not true. What has that been like?
Disappointing. And so I have suffered real career consequences, real personal consequences
Yeah.
from this. And it is very disappointing.
Should more scientists have spoken up?
Of course, of course. The core group, those who were informed and who knew in January, February, are a small fraction of scientists. But there is no scientist who could have read some of the papers that were commissioned by Fauci and Collins, and literally commissioned on a phone call, where they're confronted with scientists who are reporting to them that the scientists are concerned this had a laboratory origin and are estimating the odds of laboratory origin as 60-40 or 80-20. Those scientists were asked not to report their concerns. They were commissioned to write a paper contradicting those concerns, a paper falsely claiming that they believed there was no basis for a laboratory origin and no laboratory scenario that would be plausible.
That paper is called?
That paper is the proximal origin paper. Right. It was published in Nature Medicine in 2020. Its key authors were Christian Anderson, Robert Gary, Andrew Rambeau, and Edward Holmes.
Privately and publicly, what were they saying?
Well, we know what they were saying, and the reason we know what they were saying was that in 2023, I believe it was 2023, the House review? Yeah.
Yes.
So the reason we know is that in 2023, the United States House of Representatives Select Subcommittee on the Coronavirus Pandemic hearing resulted in the disclosure of the email correspondence and Slack rapid message correspondence of this group of authors among themselves. The correspondence shows that prior to their interaction with Fauci and Collins, prior to that emergency telephone call that Fauci scheduled on the weekend of February 1st, 2020, that they were convinced the virus likely had a lab origin.
Right.
They show
so freaking likely. Wasn't that the quote
yes,
from Christian
yes,
Anderson? Freaking likely.
That's a quote from the first author of what ultimately appeared as the proximal origin paper, right. Christian Anderson. But the correspondence proves beyond any credible doubt that this group of researchers at the start, before they began drafting their manuscript, before their call with their funding agency officials, were convinced there was a high probability of lab origin. The correspondence shows that they know the conclusions of the paper they ultimately wrote were unsound. The conclusion that it did not have a lab origin, that they knew that when they began the draft, when they completed the edited versions, when they submitted the paper, the manuscript for publication, when it was accepted for publication, when it appeared and after it appeared. At each point along the line, their correspondence shows they knew the conclusions of that manuscript were unsound, showed that they did not hold the conclusions of the manuscript, thus show that the manuscript is the product of scientific misconduct up to and including fraud. That is just the first paper, though, in a series of 4 papers. There were follow-up papers by that same core author team of scientists whose correspondence showed us that they had committed science fraud. They then went on in 2022 to publish 2 papers in the journal Science purporting to provide evidence for a market origin of SARS-CoV-2, the virus that causes COVID-19. And in 2024, to prepare a paper in the journal Cell, claiming to provide further evidence, not new evidence, but reinterpretation of evidence supporting a market origin.
Mm-hmm.
All 4 of the papers, the clearly fraudulent paper from 2020, as well as the follow-up papers, are unsound, which is to say that they do not meet the standards of science for publication in the journals in which they appear. They do not withstand scrutiny as being scientific papers by any experienced scientist. Any experienced scientist reading any of those will immediately recognize that the only commonality those 4 papers share with a scientific publication, a scientific research report, is formatting and the venue in which they were published. The content is not the content of science. The content is propaganda. The content is pseudoscience. The content is pseudoscience,
Right.
not science.
That's a name they'd hurl at us.
Yes, but other scientists, even scientists who weren't familiar with the research being performed in Wuhan before the outbreak, on reading those papers, would immediately have noticed that these papers bear no similarity to scientific research reports, would immediately have realized that something very unusual is occurring here, something that involves complicity of the researchers, their funding agencies, the scientific journals in which these papers appear, something that is very much not science and very much a cover-up appears. But until very recently, almost all have chosen to remain silent. And they've chosen to remain silent in order not to strain their ties to the organizations that fund them. That's to say, that pay their salaries. Part or all of almost every infectious disease researcher's salary in the United States is paid by Fauci's former organization, NIAID. The staff salaries for their support staff, the equipment funding, the supplies funding, all of the operating costs are provided by funding that comes from the individuals who commissioned that series of papers, namely namely Anthony Fauci and Francis Collins, up until their retirement at the end of 2022.
I've seen letters being written to universities. I mean, I've never seen anything like this in my entire life, for people speaking out about the origins of the pandemic. But
again, this group of funding agency officials were able to deploy the full resources, their multi-million-dollar budgets, and their staffs to penalize researchers who sought to expose their involvement, to reward researchers who sought to cover their involvement. They have also used those resources to win the support from scientific journals that participated complicitly, editors of scientific journals who participated complicitly, and scientific professional societies and scientific organizations and their leadership, which participated complicitly.
I think people don't understand the media's role in propagating misinformation.
The media's role is complex, but the fact that this is a science-related story, this is not a scientific story, and one does not need scientific expertise to follow this story. This is a story of human misconduct. But because there are scientific connections in this story, the story has been covered primarily by science journalists. A paper like the New York Times will assign this story to its science writers. A paper like the Washington Post will assign this to science writers. Science writers are extremely dependent on individuals like individuals like Anthony Fauci and Francis Collins. Those are their key sources. Those are their key go-to experts who provide commentary for a story and who open through introductions, avenues of approach to other scientists. And so the fact that the story is covered by science journalists and that science journalists are closely linked to the individuals at the center of this story
Yes.
created an environment where the story could be controlled effectively by those science administrators, like Fauci, like Collins. In the United Kingdom, science administrators like Jeremy Farrar, who at that time was participant in this fraud-based cover-up and who was the director of the largest funding organization for biomedical research in the United Kingdom. These individuals could use their ties with science journalists to shape scientific journalism, and the broader media took their message from this set of conflicted science journalists.
Do you consider SARS-CoV-2 to be a bioweapon?
Well, so here we need a distinction of terms. So a biological weapons agent, that's a term that I've used consistently here, or bioweapon agent, is a pathogen or biological toxin with potential to be used as a biological weapon. There is no dispute that SARS-CoV-2, like SARS-CoV-1, is a biological weapons agent. The research on SARS-related viruses being performed at the University of North Carolina, Chapel Hill, and the research being performed in Wuhan were funded by the biodefense subcategory of the NIAID budget. This is a matter of public record. One can consult the NIH records for the grants, and the spending category, the primary spending category for the research, is biodefense. These are agents that fall within those categorized high priority bioweapons agents that are eligible for prioritized biodefense funding. So the research funding on SARS-related viruses, the research involving virus discovery in the field in China, the research involving virus enhancement in the laboratory in China and in the United States, that was biological weapons agents research that was prioritized and funded expressly and explicitly as biodefense research. Those pathogens are biological weapons agents. You asked about bioweapons. Bioweapons, without the word agent, refers to a biological weapons agent that is produced for use as a weapon. So the difference between a biological weapons agent and a bioweapon is intent. So if you are studying the agent, it is an agent, a biological weapons agent. If you are now producing and packaging the agent for distribution as a weapon, it is a bioweapon. SARS-CoV-2 was a biological weapons agent.
Have you gotten SARS-CoV-2? Have you been infected?
No.
So
I'm a COVID virgin.
You and me both. I think so anyway. And I stand in solidarity with you. You know, much has been made about us last holdouts. And I want to know from you as an expert on this agent and just in this field, why do you still protect yourself from SARS-CoV-2?
It is clear that a subset of persons who become infected have long-term consequences. Yes. Long COVID, chronic COVID. Not the majority, but a subset. If I can protect myself from infection at minimal risk and minimal cost and minimal effort and avoid those consequences or the possibility of those consequences, I would be foolish not to.
I can't argue with that. And yet here we are where being a person that's still protecting themselves is considered fringe, crazy, bizarre. Didn't you get the memo that this thing is over? And so I try to explain it like that the initial infection does not tell you what's going to happen down the road. And as Dr. Redfield very eloquently explained, it's a blood vessel disease. And you can't feel your blood vessels. You don't really know what's going on.
It comes down again to an assessment of risks and benefits.
Yeah.
The discussion of whether the research that caused this pandemic should have been performed, one can assess risks and benefits for oneself. And the kinds of risks that we faced in 2020, we no longer face in 2024.
Yeah.
And the benefits of precautions that made sense in 2020 no longer make sense in 2024. But masking when in public, indoors, the costs are very low. The benefits are significant.
Do you eat in restaurants?
Rarely. But I did as recently as this weekend. And the mask was off indoors while eating.
Would you take antivirals if you got infected? And do you think they're important?
I would, yes. I would take Paxlovid.
Did we cover all the important? I had an
so.
enormous outline,
Yes.
many questions. And of course, I can't let you go without wishing you a happy Thanksgiving.
You
And
too?
I need to know, are you going to do the chicken this year? Or is it
Yes.
going to be the
Yes.
famous Costco chicken?
Yes. It has been purchased
I love it.
today.
Well,
I heard
okay.
it arrive in the background while we were speaking.
That's fabulous. Well, I wish you and your family a very happy and healthy Thanksgiving and year, and I really appreciated your time today. Thank you.
Likewise for you, Dana.
Thanks for joining us today. Please subscribe or follow along for more really interesting conversations, and I'd love to hear from you. So please drop a comment or send me a note. Let me know what you think.